Thank you very much to the Martinez Center and Viviana for inviting me to give this talk. So I'll be talking about the retrospective and prospective harmonization of diffusion MRI data. And okay, so let's see what the next roughly 1 hour looks like. So why harmonization? I'm going to start with that and kind of provide some perspective on what I mean by harmonization and how is it different than data pooling. And then I'll present some of our recent work on retrospective harmonization as well as prospective harmonization. And then I'll sort of end that talk with some challenges and open questions both in harmonizations and data pooling. So reproducibility, I think everybody sort of knows about, you know, the lack of reproducibility in many studies. Part of the problem probably lies with data acquired on multiple scanners. But let's see what reproducibility looks like for the same scanner and the same subject. And so what if we were to scan the same subject on the same scanner, multiple sessions? We're talking about, I'll mostly talk about diffusion MRI here, and this is what you see here on the left. Percentage change in FA for the same subject, very small variability. Basically, it's about 2 to 4% in the white matter, maybe 10% in places in the gray matter. And on the right also, you see a plot where 3 subjects were scanned on the same scanner, multiple sessions, and at a whole brain white matter level, you see pretty much very consistent sort of data. So you can, you know, assume that about 2 to 4% in white matter is what the changes are, if you were to look at it from that perspective. But even this change, why do we see these changes? Why aren't the numbers exactly the same? Well, we have differences in shimming when you scan and rescan the same subject in different sessions. There is also biology-induced variation, so pulsation effects can be different. Also, there is some recent work to show that the time of day can impact the measures, especially for diffusion. So, you know, if you scan in the morning versus in the afternoon or evening, you might be able to see some differences there. Also, gradient performance variations, wear and tear might affect these minor variabilities. But now moving from within scanner to inter-scanner variability. So how much differences are we expecting here? So here is you know, a coronal slice. What we see is the same subject scan on 2 different scanners, Siemens versus GE, and we're showing the percentage change in FA. And as you can see, white matter, obviously there is less variability, but yet it is 10% at least, maybe more in the white-gray interface or boundary, and it's about maybe 20% in the gray matter and much more in the CSF areas. And this is also seen in the plot from the right, where you see that the same subject scanned on 2 different scanners show very large change in what we measure for generalized fractional anisotropy here. And it's not 1 subject here, there is 3 subjects. You can see pretty much consistent set of changes. So why is it important to, you know, think about this? You know, 10–20% change, maybe not that much, but really speaking, the reason this is important is because when we're looking in neuroimaging studies, what you're expecting is that between healthy and any type of, say, psychiatric disorder, you expect at a group level, a statistical group level, the changes are about 5%. So here are 2 examples. On the left, you see changes in FA and radial diffusivity between 2 groups, about 5% change. On the right is another paper from our group. Again, here you're looking at differences between 2 groups. One is soccer players, another is set of swimmers. even though the groups are very well separated, yet if you look at the percentage change, it's just about 14%. So it's extremely important that we reduce these inter-scanner changes. You can imagine that if these subjects were to come from a different scanner, two different scanners, you would probably almost wash out all the differences between the groups. And this is what you can see in this plot as well. If you were to not harmonize the data, and here we have data sets acquired from 3 different scanners shown here in the dotted line. And if you were to just take them and analyze them, here we are analyzing or looking at the lifespan change in fractional anisotropy in male healthy subjects over age, you will get something which is, very, very potentially erroneous. Whereas if you harmonize, you see the trajectory that is very similar to what we have seen if you were to get all of these subjects from the same scanner. So very important that we harmonize the data from different scanners. Now, where do these differences between the scanners come from? And one is, the magnetic field inhomogeneities. There are differences between scanners on them. And then there are many other contributors to it also. You might have different sensitivity of the coils, the receiver coils, as well as the number of the receiver coils. So one site might have 16, another might have 20, another 32, and so on. Then you have differences due to the reconstruction algorithms themselves. For example, if you were to go on a Siemens scanner, it probably ends up using a Grappa reconstruction. On a different scanner, it might be a Sense reconstruction. And then sometimes people also differ in the type of partial Fourier imaging that they do. So these all contribute to differences in the scanner, in the data that you obtain from different scanners. And these differences manifest themselves to be regionally dependent. They are also tissue specific. So, you see, as you can see on the right, the gray matter, the white matter, the subcortical gray matter have very different types of differences in FA here. And as I will show along in the rest of the talk, these differences also present in all frequency ranges in the diffusion MRI signal. So it's not just a constant change, as you can see from the plot on the right. And as a result, these changes are nonlinear. So they're not linear changes in the signal itself. And that's one of the reasons why physical phantoms may be too simplistic, yet the way the physical phantoms are available are maybe too simplistic to be used for data harmonizations, because you don't know how to translate these changes in the phantom to a particular change in the brain, the human brain. And so we actually end up needing human phantoms. So I guess I've convinced you about why there are changes and why harmonization needs to be done. But apart from the fact that there are differences, if you harmonize the data, you end up increasing the statistical power. And of course, now you can combine data sets and answer questions that you wouldn't be able to answer otherwise. And so the goal of harmonization in broad terms to me is to reduce the interscanner variability in the dMRI data to something that is very similar to an intrascanner variability, so that scanner specific biases are removed. And ensuring, while doing this, we need to ensure that all possible information in the signal is preserved, that the biological variability at the scan sites are preserved. And you also want to make sure that when you're doing these harmonization, that the fiber orientations are preserved when you move the data from, or when you harmonize the data from one side to the other. So there have been, and this problem was known quite some time ago, and people have looked into it, not only for diffusion MRI, but also for structural MRI. But in this case, for diffusion MRI, people have used statistical methods, and I'm going to go over a few of them that have been used in the past. So you have meta and mega analysis methods. So people might be very familiar with the Enigma consortium, the software. They have been able to combine large, very, very large number of data sets from around the world. And that shows the success of the use of these analysis techniques. The way it works out is that you calculate a z-score at each site and for each of the diffusion measures of interest, and then you combine those z-scores together. So instead of you looking at the data diffusion measures themselves, you're looking at the z-scores after you've computed them at each site. Potential confound for that is just that if your sample size is small in one of the sites, then you're not actually capturing the entire biological variability, and the z-score depends on this particular factor. So you have the standard deviation of the sample in computing the z-score, and if your variability is not properly captured, your z-scores could be completely off. And so sampling bias can create some problems there. And obviously this assumes that there is the changes between the scanners are all linear within the entire region being analyzed. And so this method is really great if sites cannot share the raw data, or that they can only share some of the partial data, and because of various restrictions. So this really works out well in that particular scenario, and very easy and fast. Then there is method to add statistical covariates. So in this case, you add a covariate for site at each voxel. And this obviously has the advantage that it captures the regional and tissue specific changes, differences in the sites. But again, it assumes everything is linear, and that which may or may not be true in many cases. Then there is another method that has been proposed, which is to use a correction factor. So in this case, the authors used 3 traveling subjects from multiple sites, and they had calculate the measure of interest in a ROI of interest, for example, FA, and then they find what is the correction factor, and then they add that correction factor to everybody else in the data set. This works well as long as the correction factor properly captures everything. But confounds can exist if you are looking at large ROIs. For example, the corticospinal tract. You know, you're going from the base of the brain to all the way to the cortical regions, and along the path, these changes may not be linear. And also the question is, what happens to your estimation algorithms, your tractography algorithms downstream if you are only harmonizing at the very end of the pipeline? Then there has been recent work for a method called COMBAT, which has been very successful in the genetics field, has been borrowed and applied to diffusion and structural MRI as well. In this case, you use a voxel-wise additive and multiplicative factor to account for site effects. So apart from just using an additive factor, you also have a multiplicative factor. There are certain assumptions made about what the distribution of these factors can be, and these need to be estimated when you do the analysis. And we started to look into this for over 5 years ago now, where we looked at this not at the very end of the pipeline, but to harmonize the signal, the raw signal cut of the scanner itself. And in this case, you could use either traveling subjects or match controls to learn a mapping between the 2 sites on the raw data itself. And this is voxel specific, so regional and tissue effects, tissue specific effects are also captured. And it looks at non-linearities in different frequencies. And so, you know, it accounts for all of those as well. There is one particular thing which is a requirement for this method, is that it requires a matched set of controls or traveling subjects to learn this intersite mapping. And there have been, since then, there has been a huge growth in the number of, in the field, where people have started to look at various ways to use this from raw diffusion MRI based method for harmonization. And as you can see, these sort of publications have started to come out in the past year or two. So to provide some brief comparison between what I call data pooling are basically methods that use statistical covariates or statistical methods of meta or mega analysis to account for scanner differences. They are done at the very end of the pipeline. And done for each diffusion measure of interest separately. And most of these methods that I talked about regarding statistical analysis fall under this category, where at the very end, you run through your entire pipeline, and at the very end, you add this extra statistical covariate to account for differences in the scanner. On the other hand, harmonization, we think that it is it is done at the very beginning of the pipeline after pre-processing. So very first step, applied to the raw data itself. So this has the advantage that you can do apply any type of model to the data you want, and you don't need to worry about it. And it's it can be linear or nonlinear mapping, so you can learn all the nonlinearities in the data itself when you perform the harmonization. So a few things about data pooling that we have also started to look a little bit more deeper into, but still many unknown factors yet. So for example, it's unknown how scanner-related nonlinearities propagate through the processing pipeline, because all the processing is done and then you account for scanner-related things. We don't know how these intrinsic differences in the signal affect and their interplay with the processing pipelines. I'll give one example of that going forward. Then also, we don't know how the modeling assumptions, how do they affect the harmonization or statistical analysis. And we also don't know what is the effect of noise characteristics from different scanners. There might be slightly different noise characteristics, and how do they play out for diffusion measures and during harmonization. So here is an example of what we recently did. What we see here is the effect of software processing pipeline on a set of data sets. Here we have is 5 different sites. We took about, I think, about 400 data sets, each probably having something like 40 to 50 subjects. We pre-processed all of the data sets exactly the same way. And then we passed them through 3 different softwares. So one is FSL, another is MRTrix, and 3 is Slicer. So you have 3 different popular software packages that were used to run ordinary least squares diffusion tensor fitting. Very simple. Calculated the whole brain mean FA, which is across the entire brain. And naturally, this is entire brain, but what we see is that for sites 1, 2, 4, 5, pretty much everybody performs the same. But very interesting was what we see in site number 3, where you see statistical differences between the 3 softwares. So this data is exactly the same, just that the fitting and whatever else is done in the software was different. And you can see very large amount of differences here. Now, we also did something with Combat, which I want to present here, where we try to use Combat for harmonization. Here you can see what we are plotting here in this bar graph is effect sizes between healthy controls and schizophrenia patients. You have effect sizes before harmonization, so between controls and patients. And then you have in the orange after harmonization, but using the same software. So the same software pipeline was used. And if you happen to use different softwares to process, then what happens? So for site 2, you can see, and what you see on here for each of the bar graphs is different areas of the white matter. you see for site 2, pretty much everything behaves as we think it should, which is that before and after harmonization, the effect sizes pretty much stay the same. But things become a little bit different for site 3. So here you can see that if you process the data with the same processing stream, the before and after harmonization kind of stays the same, which means the effect sizes are pretty much the same. But it's very different if you processed it to different pipelines. And in some cases, you can see here the effect sizes, initially were 0.5 or 0.4, and now they're 0.9. Things like these. So you're suddenly, have a very different effect size between the groups, which you shouldn't be having after harmonization. You need to preserve the effect sizes. It's a different example here. What we see is the effect size between males and females healthy subjects here. Again, the same set of analysis. We have before harmonization, after harmonization, 2 different categories, same software, different software. Now here, site 1 again looks pretty good. Things look very, very nice and very, very much as expected. But in site 5, what you start to see is very different. Here you see at the whole brain level even, you see that before harmonization, the effect size was about -0.2. Now, even if you're using the same software, after harmonization, your effect size is now -0.5. And in some cases, you can see here, the effect sizes have flipped the direction. So what it means is that, in this case, if the females were having a higher FA than male, now after harmonization, it's the opposite. And this is at a group level. So there could be certain issues that might be affecting how combat works, because these nonlinearities in processing the data has perhaps added more to the problems for harmonization in certain cases. As we can see, for site 1, it works, but site 5, it doesn't. So that's the motivation to try to harmonize the signal at an early stage. So we suggest that we should harmonize it at the earliest possible stage and harmonize the signal itself and not the diffusion measures. So I'm now going to give a brief overview of the work that we did, and more or less, it starts with you have site 1, site 2. You either have controls at both sites, matched set of controls, or you have traveling subjects from both sides, and you want to learn a nonlinear mapping between the 2. And the way we go about doing it is that we first pre-process the data. So you need to ensure that you do your eddy current correction, your motion correction, your Gibbs unringing, EPI distortion correction. And in certain cases, you might have differences in spatial resolution. You might want to match them. Then we compute what is called the Riesz features, or rotation invariant spherical harmonic features. And I'm going to go step by step in detail for each of these steps in the upcoming slides. But in general, these are features derived from the diffusion MRI signal, and kind of compactly and succinctly represent the data very well. And they are also rotation invariant, so very nice feature to have. Then you align all of these data sets from all the sites into a single template space. Then at a group level, you learn a statistical mapping between the 2 groups. So for the Riesz features, because this is the domain in which we work. And then once you know this mapping, you update the signal. Because there is a 1-to-1 mapping, you can go back and update the signal based on the spherical harmonic representation. And so, as I mentioned, we need to first do our pre-processing just to make sure everything, all the other factors and artifacts are taken care of. In case you have differences in the spatial resolution, we have used a 7th order B-spline to fit the data, to interpolate the data. And this was based on work from Derby et al., where they have shown that the 7th order B-spline gives very good results comparable to what you would acquire in vivo. So you can use them very robustly. and we remove the Gibbs artifact. And also for retrospective harmonization, if there are differences in B value within a certain range, and we can definitely talk about this, but in a very small range, you can match them very easily, assuming some linearity between them. And the way it works out is, for example, you take a voxel in the brain, and this represents a signal. You represent the signal in a spherical harmonic basis. This is the pictorial view of the basis. So you start from a lower order and all the way up to higher order frequencies. And once you fit it, you get certain coefficients for each of these basis functions. And essentially, then you calculate what are Risch features, which is basically the rotation invariant features. So you take the first coefficient and basically the norm of that forms the 0th order Risch feature. Then you take all the second order coefficients and take the norm, and that forms the second order Risch feature, and so on. The odd harmonics are typically 0 because our signal is symmetric. And so you compute these features at each voxel and you get some feature maps. And as you can see, these are the feature maps. This is averaged over a large number of subjects. So these spherical harmonics are great representations. They are general, they are orthogonal basis functions, and rotation invariance is a great feature. And it also shows what you see here is all the different orders. So different orders capture different frequencies, and you can see that there are differences in the brain regions as to what is more relevant compared to one versus the other. So different orders captures different parts of the diffusion signal spectrum. And so in the retrospective case, one important assumption that I would like to point out with our method that you need to be aware of when we do any kind of harmonization, but specifically in this case, we assume that two groups of well-matched healthy controls will not show statistical differences. So if you have well-matched set of controls, and this is the case for retrospective data, because you don't have any other way to know to match the two sites, you only can match them based on controls. So you match them from each of the sites based on age, sex, IQ. If proper matching is not possible, you will have to regress out the confounding variables. But the best case scenario would be to match the controls from the two sites. And then you perform spatial normalization or registration, as I mentioned, for all subjects and all sites together. And then you learn a linear or a nonlinear mapping between the group averages of the Risch features from each site. So at a voxel level, you take the group average because you have registered all of them in the same space. You can take the group average at that location for both the sites. And let's say you can learn a very linear map. Let's say linear mapping would be how much percentage difference or scaling factor is needed for each of the Risch features. So I would emphasize that this is done for each of the Risch features, which means that you're looking at a decomposition of the signal in the spherical harmonic domain, and you're matching in that domain. And then you once you know this, you can go back and update your dMRI signal at the target site. And this is what I essentially talked about. This is just a set of equations, but pretty much the only slide with equations here. So here you have this signal represented in a spherical harmonic basis. The CLM are your spherical harmonic coefficients. And this is a scale map that you learned for the Risch features as the ratio between the Risch features from the two scanners, the group average Risch feature ratio. And so let's say this is the scale, and then you update the coefficients using the scale, and you get the new coefficients. And once you know the new coefficients, this YLM stands for the spherical harmonic basis. So once you know the new coefficients, you can compute back what the signal would look like after harmonization. And so here is an example. So on the top are we have in this case two data sets. One is the Philadelphia neurodevelopmental cohort, the PNC data. There is the Brigham and Women's data set. On the top, you see both of them were having 24 subjects match very well for each of the sites for age, sex, IQ, etc. And on the top is the average for the group, average for each of the reach features. And you can see the PNC and the BWH have very different looking reach features. And this is the scale map that we obtain. So, very large, differences at the 0th order, and the energy kind of goes down. But as you can see, what is important is that it's regionally specific. It depends on the region, as well as tissue and tissue specific as well, plus different for the different frequencies. These data sets had slight differences in spatial resolution, b-value, number of gradient directions, etc. So the question is, how well did we do? I mean, when we did this harmonization, how well did we do on the training data itself? So we did 2 different tests. This is a generalized fractional anisotropy when the PNC was the reference site and the BWH was the target site, which means that the BWH was harmonized. And you can see the bar graphs for different regions of the white matter. Before and after harmonization, they look pretty much the same. And this is the opposite of, in this particular case, we harmonized the BWH to the PNC site, and we got pretty much very similar looking results, which means that before and after harmonization, the effect sizes or the GFA values here in this case were pretty much the same in all of these regions. Now, what about testing on other test data sets? So we did multiple tests in this case. So here is the effect size for a variety of different white matter regions. And this is effect size comparison for, we took 2 groups who were different by age. And here, what you see is that, you see the effect size before and after harmonization in gray. So the effect sizes pretty much follow the same trend, which means that before and after harmonization, the effect sizes haven't changed much. There are minor differences, but they're really very close. And another example here, we did it in females. Again, age differences between the 2 groups exist, and we calculated the effect sizes before and after harmonization in a number of these different white matter regions. So to show comparison with combat, on here we have the age trajectory for 2 different white matter bundles, the left IFO and the left SLF. On the top is using the reach feature based method, and on the bottom is using combat. Now here we did exactly what was recommended, and what we see is that the trajectory has changed after harmonization. And this is slightly an undesirable result, but this is what you see. Because there is a nonlinearity in the FA value over age range, whereas you assume that in combat, you assume linearity of this dependent variable. So this can cause some problems here. So an important question for harmonization with this, when you do this raw diffusion harmonization, is how many well-matched training subjects do you need from each site for accurate mapping? And this is what we did here in this plots. These plots show you how the average, for example, the scale map that we calculate, what is the average when you keep increasing the number of training data sets. And as you can see, these numbers kind of converge to a certain point, and pretty much about 14 or more subjects are required for well-matched subjects are required for learning this mapping between the 2 sites using this linear maps. And we tested this with multiple different data sets, and more results are available in the paper. To show this more pictorially, this is the error between the scale map when you use different number of training data sets. So what you see is for each column is the training data set, different number of training data sets. and each row is a different reach feature or a different frequency of the spherical harmonic fit. And what you see is that if you have only 2 training data sets, obviously the error is very large. But as you go about going to about 16, you see that the error pretty much, 16 and 18, the error between the final fit and the scale map error with only using 18 is pretty close. It is pretty close. So now moving on to prospective harmonization. In this case, what I mean by prospective harmonization is that you're going to start a study. You have the opportunity to design the whole study for multi-site or multi-center studies, and you could potentially have the possibility of using traveling subjects. In this case, how do you go about doing this? Until now, I was talking about retrospective, where you don't have this option, which means that the data has already been acquired. There is nothing else you can do about it. So in prospective harmonization, one possibility is to use physical phantoms, and we'll go over it again. You can also, you definitely should use traveling subjects acquired at multiple sites, and then you learn a mapping between them using these traveling subjects. And then apply this map that you learned to the remaining data sets. And this is what you want. This is data from, where we had the traveling subject go from one side to the other. And this is before and after harmonization, percentage change in FA, as you see after harmonization. Again, this is what is the desired result with very little change after. So for phantoms, definitely physical phantoms are a must in any type of multi-center study, and not just multi-center study, any type of long-term study where you want to monitor any type of software upgrade or drift in the scanner or any other artifact that might be caused. There was a very nice study done recently where the authors used a single strand GTI phantom, and they showed that almost all scanner variables, and this is in Europe, where they scanned this single strand GTI phantom across, I think, about 13 or 14 different sites and multiple times over a period of 2 years. And what they showed was that almost all scanner variables contribute to differences in the acquired data. So whether it's coils, scanner drift, bandwidth, software vendor, site, all of these variables caused a difference. You can quantify these differences using a mixed effects models or any other model. But the question really is, how can we translate what you know from this phantom, where you calculated the FA and the mean diffusivity, to differences in the human brain? How do you map them? And that's pretty non-trivial. Because we know that it's regionally specific and dependent on tissue and other nonlinear effects. So another next best option is to use human phantoms. And what I'm showing here is we use the same method that I described earlier, where we calculated a map between 2 sites where we had the same set of subjects scanned on both sites. And what we see here is before and after harmonization, as you can see. And this is what is demonstrating is the mean squared difference in the dMRI signal itself across all different gradient directions. What you can see is that at least inside in the brain parenchyma, after harmonization, the differences are very well removed. We also did some other experiments as well, where very interesting data sets were available to us. For example, we thought, what about the extreme case of harmonizing data between sites when there are very, very different magnetic strengths? So 3 Tesla and 7 Tesla data sets. Can we even think about doing harmonization in this case? And turned out that we had access to a very beautiful data set, about 150 subjects scanned on the 3 Tesla connectome and the 7 Tesla scanner as part of the Human Project, HCP project. And this is a large enough pool to figure out whether this can even be done. And so we went about using a deep learning method where we used a convolution neural network here, where the input was the 3 Tesla data at B values of 1000 and 2000, and the output was a matched set of 7 Tesla data, 1000 and 2000 D value. We use 9 by 9 by 9 paired matches, and then we run through the convolutional neural network. And what we found was that what this plot is showing you is again how many data sets were needed to learn this nonlinear mapping. And because this is patch based, we used about 1000 patches per subject. And so what you see is that on the validation set, a plateau starts to occur at about 10,000 patches or about 10 subjects. So what we needed was at least 10 subjects to train using this particular deep CNN model. And what about the results? So here is what I'm showing is the test results on 60 test subjects. So we just learned the data from 20 subjects and then tested them on totally unknown 60 test subjects, which were obviously not in the training. And what you see here is that the original data set, what I'm showing you, these numbers are the percentage difference between the original 3T and the 7 Tesla data in the root mean squared error, percentage error. In FA, mean diffusivity, generalized fractional anisotropy, and diffusion weighted images themselves. It's about 30-35% differences between them. Using just the linear method that we talked about earlier, the differences dramatically go down, but yet not there potentially. Then we use the deep CNN, which I mentioned earlier. And then finally, another method which we used was cycle GAN, which is another form of deep learning method, which is generative adversarial network. And here you can see that at least in the FA, the root mean squared error is about 5%, in MD it is 2%, and so on. And it is, as I mentioned very early on, the differences for the within scanner differences are up 4%. And I think we are getting closer to the limit, obviously not there yet, but still getting closer. And I think this is promising this looks promising basically. These are some of the difference maps, which is definitely important to see because what you can see is that the difference map in our percentage difference in FA. And while you can see some structure in the linear method, with the deep CNN, it is pretty much very little. And with this new cycle GAN, this essentially the difference map at the bottom right here is looks to be pretty much noise. There is not much of a structure there, but as you can see. So then the question is, okay, we looked at FA and mean diffusivity. What about fiber orientations? As I talked earlier, are they changed when we do this harmonization? So we looked at several different fiber bundles. A couple of them are shown here, the cingulum bundle, the corticospinal tract. And what we see is that there is a significant overlap between the fiber bundles. We tested 4 or 5 of them. And you see about 95% or more sort of overlap between the tracks, which is encouraging. Given the importance of this harmonization, there has been a couple of different challenges at MICHI. One of them was the recent MUSHAC challenge, where the challenge organizers acquired 15 traveling subjects from 2 different scanners, Prisma and Connectome. They provided us 10 for training, and 5 were provided for testing, which means that they only provide one of the scans. The Prisma scans were provided, and we had to guess based on the 10 training data sets what the remaining, what the 5 test Connectome scanner data would look like. And so it was a blind challenge. 21 different teams participated in the challenge. And one of this paper is out and shows how well our method worked here. What we see here is that the absolute percentage error in FA and mean kurtosis. Mean kurtosis is again another measure which is using higher B values. Here you can see that our method is doing very well compared to all the other sort of methods that participated in the challenge. So to sort of getting to the end of the talk here, so there is what we feel is that there is obviously still work to be done, but despite differences in T1, T2, echo time, relaxation, and magnetic field inhomogeneities, there is a possibility to learn or to learn a mapping between sites, even if there are large differences. So this also was, as I said, this was an extreme case, but this also provides hope that with linear and maybe potentially nonlinear deep learning type of for traditional 3D scanners where most of the data has been acquired, there is a possibility to reduce these inter-scan differences to something that looks like intra-scanner differences. And so our focus right now is on that. Our current directions are that we would like, we are harmonizing 10,000 data set, which is a 40 site sub data. Is done in a retrospective manner. There are also 10,000 subjects from the HCP and the disease connectome, as well as 10,000 subjects from the healthy brain network, which will be a prospective study, harmonization where traveling subjects will be available. And all of this data, once harmonized, will be available on the NIMH data archive. All of that study is funded as part of our R1 grant. And very briefly about this grant, if people are interested, our first step would be harmonization of the grant of the data and deposit it to the NIMH data archive. The aim too would be to do tractography and extract more than 150 to about 190 white matter fiber bundles, well annotated based on known human neuroanatomy. And then we will also provide a visualization tool for people to explore and mine this data from the NIMH data archive itself. So to summarize, we think that harmonizing the signal at the earliest possible stage is probably better because you avoid a lot of the problems with during the processing streams. It is also model independent, so any type of analysis can be used in the future. So you could use single tensor, multi-tensor, DSI, any other model. And for prospective multi-center studies, use of traveling subjects, this method allows you to use that as well, whereas some of the statistical methods will not be possible to do that. And also tractography can be done in a consistent manner, so that because after harmonization, the signal is looking very similar, as well as some of the noise characteristics have been muted. Some of the caveats with harmonization method that I would like to point out. So when we do this harmonization, any method, we want to ensure that we are not introducing any new information in the data as a process of harmonization. So for example, any new peaks or change in the fiber orientations. Also want to make sure that you validate, that we validate the harmonization using model independent metrics on the raw signal itself, as well as some very widely used ones. And it's also important to characterize the effect of noise distribution on the harmonization technique. Some open questions regarding data pooling. I would say that certain things to keep in mind and give a thought to before you do this retrospective data pooling, use this data pooling techniques. What if the data sets have very different b-values? We know that the b-value and the FA and mean diffusivity are not linear. They don't vary linearly. So there is a nonlinear relationship there. How does the statistical adjustment work in this case? What about spatial resolution? So if you have two data sets with very different spatial resolution, partial volume effects will be an issue. And bias, there could be bias due to nonlinear site effects as well. We need to be very cognitive of the fact that when you do mega analysis, for example, if one of the sites has a very small control set of subjects, then do you end up artificially enhancing the group differences between them? And there could be also potential bias in FA if if a measure of interest, like for example, FA varies in a nonlinear fashion with certain of these clinical variables like age, IQ, etc. And so we need to know all of these things are what the assumptions are before we use these techniques, whether it's harmonization or data pooling. And pretty much getting to the last few slides here. Next few set of directions for us. We are, what about gross pathology? What happens there? Still something to be figured out. In prospective harmonization, the question is, how can we capture scanner differences with as few traveling subjects as possible? And will this be consistent across vendors and versions of MR software? What about inter-protocol differences? So I think that's another interesting question that we are looking to answer is, if you have been acquiring data with a particular protocol and now there is an advanced protocol and you want to use that for the rest of the data set, can we do some sort of harmonization there? Obviously, there will be some limitations. And I think if we can, we might be getting closer in this case if we can achieve these sort of harmonization that could be used potentially in clinical trials for several pathologies. Also looking into structure and function MRI data. These are some of the relevant publications if folks are interested to know about them. And I would like to thank all my collaborators who have helped in this project. Special thanks to Sohaila, who actually did all of this work. And so I'm happy to answer questions. People are interested in looking at using our harmonization software. It's publicly available here at the GitHub page given. And thank you very much. I'm happy to answer questions now.